Hello everyone, I am Dr. VC Manoj, President of National Neonatology Forum Kerala and Organizing Chair of IAP Neocon 2021. I have great pleasure in welcoming you all to today's the 13th session in our International Neonatology webinar series. It's a humbling experience for us to have so many attendees for this session from 65 countries all around the world. Welcome dear friends to yet another Learn from the Legends webinar. We have with us today two eminent neonatologists of India, Professor Dr. Kumuda and Professor Dr. Chandragala, to moderate today's session. Hearty welcome to you, madams. And now let me welcome
Good evening, Dr. Manoj.
Good evening, madam. Now let me welcome the legend of the day, Professor Joseph New. Dr. Joseph New did his medical school training at University of Wisconsin, was a pediatric resident at Johns Hopkins, and a postdoctoral neonatology fellow at Stanford University. He came to the University of Florida as an associate professor in 1984 to continue his research in developmental gastroenterology and neonatal biochemical nutrition. In 1987, he pursued additional research training at the University of Bern, Switzerland, on the basic mechanisms affecting intracellular processing of lactase in intestinal epithelium. He is now internationally recognized for his research in developmental gastroenterology and nutrition, and has most recently focused his research on efforts on the intestinal microbiome and mucosal immunology. Professor New has been NIH funded to study the developing microbiome in a babies at risk for developing necrotizing enterocolitis. This involves a multi-center evaluation of intestinal microbiota using novel non-culture based technologies. He is also evaluating the effects of the fetal microbiome as it relates to prematurity and evaluating the effects of antibiotics on the fetal microbiome and metabolome of preterm neonates. Dr. New maintains an active interest in residency and fellowship training as well. It's not just research alone. He has been involved in direct mentorship of over 20 postdoctoral MD neonatology fellows, 3 pediatric gastroenterology fellows, approximately 15 visiting international scholars, 3 PhD postdoctoral fellows, and over 20 undergraduate independent study students at any time, the majority of whom have gone on to medical, veterinary, or graduate school in the sciences. He has also mentored 15 high school students in his laboratory. If I keep reading, it will take more than an hour, so I will just cut short. He has served as the director of neonatology fellowship training program at University of Florida since 1986, and is on the Council for Organization Neonatal Training Program Directors (ONTPD), and served as the national chairman of the ONTPD for 2 years. He is on the editorial board of 7 journals, has served on numerous NIH study sections, and is also currently the chairman of the board of the Hippocrates Foundation. Well, friends, some of us must be recollecting the great Hippocrates seminars which we must have attended in various parts of the world. For those of you who don't know about Hippocrates, Hippocrates stands for International Postgraduate Organization for Knowledge Transfer, Research and Teaching Excellence Students. Professor New has organized various seminars in neonatal gastroenterology and nutrition in numerous countries of the world. He is now the chairman of the board of Hippocrates Foundation. Well, microbiome, gut-brain axis and enteric nervous system, epigenetics and more. We know it's a fascinating world out there. Let us now hear the legend talk to us on this amazing world of microbiomics, genomics, proteomics, metabolomics, and beyond. The multi-omics approach in the pregnant women, fetus, and neonate. Welcome, Professor Joseph New. Over to you, sir.
Thank you, Dr. Manoj. It's a real honor to be involved here. And good morning, good afternoon, good evening to whoever, whatever your time zone is. And I hope that you enjoy this lecture. So I will be talking about multi-omic approaches in the pregnant woman, fetus, and newborn. And just for housekeeping purposes, this is my disclosure statement. I have no conflicts of interest to disclose here. Over the next 50 or so minutes, I'm going to provide some historical perspectives of the microbiome primarily. I'll talk about the microbiome and early life intestinal microbial interactions, environmental factors affecting microbial intestinal interactions, and I will focus on antibiotics in preterm newborns. And then I'll talk about another term called multi-omics and why that's important, and then we'll have some clinical correlations. So let's begin here. At the top of this slide, you see this individual who is collecting a sample of water from a body of water.
So please share the slides, sir.
Oh, I'm sorry. I thought I had that. Okay. Okay, let me move back here. So can you see this okay now?
Yes,
Yeah.
we can see you.
Okay, excellent. Okay. So here's this individual who is collecting a sample of water. This could be from a river, from an ocean, and even if you consider that sample of water to be very, very clean, if you put that sample of water under a microscope, you are very likely to see many microorganisms. But if you take that same sample of water and put it onto a petri dish, and with that petri dish, you try to culture these microbes with regular culture media, you will probably be very unlikely to be able to culture all of the different microbes present in that water sample. And this is something that has become called the Great Plate Count anomaly. So this means that there are many, many cells that cannot be, or many microbial cells that cannot be cultured. Now, over the past 2 or so decades, the story has become a little bit different. As you all know, there was this very large project, worldwide project called the Human Genome Project. And with the Human Genome Project, that started in the late 1980s, early 1990s, the purpose was to sequence the adenine, thymine, guanine, and cytosine in human DNA. And with a cost of over $3 billion and over 10 years of major effort, this was done. The genetic code was cracked. And here are just some of the publications that resulted from that cracking of the genetic code. Now, along with the cracking of the genetic code, new technologies were developed, and these technologies involved primarily sequencing and bioinformatics. And here on this slide, we see the application of some of these technologies to microbial DNA sequencing. And here we see two types of technologies. One is called 16S ribosomal RNA sequencing, and the other is called total microbiome DNA sequencing. The RNA sequencing, the ribosomal RNA sequencing, involves just taking a small area of the microbial DNA and sequencing that particular region, and then comparing that with some known sequences. And that will allow you to determine who is there. In other words, what the microbial taxonomy is in a certain niche or in a certain sample. You can get a lot more information by looking at the total microbiome DNA, and that's looking at the entire sequence of all of the DNA in that sample. And by doing this, you will get a lot more information in terms of what is the capability, what are the genetic capabilities of the microbes present in that particular sample. So here you get the taxonomy as well as what they are doing, what their potential function is. This whole genome sequencing is still quite a bit more expensive than the 16S sequencing, but the prices are coming down markedly. So 16S, who's there? Whole genome sequencing, what are they actually doing? In 2007, the Human Microbiome Project started. And the goal of this was to improve the understanding of the microbial flora involved in human health and disease. And since that time, we've come a long ways. And I'm going to focus primarily in this lecture on the pregnant women, the fetus, and the newborn. Now, in terms of this Human Microbiome Project, using some of the techniques that I just showed you, it was possible to determine that in different niches of the body, the lung, the gastrointestinal tract, the skin, the urogenital tract, there exist numerous species of microorganisms. So in the gastrointestinal tract, there are thought to be 500 to 1,000 species. On the skin, 1,000 species. In the lungs, there are around 600 species. Many of these were never cultivatable in the past, but now with these new technologies, we are beginning to recognize that there are many more microbes present and that they are potentially very important to our health and well-being. In the gastrointestinal tract, which I'm going to focus on here, we are beginning to recognize that it's more than just a digestive absorptive organ. It's also the largest immune organ of the body. It harbors the enteric nervous system. In fact, there are as many nervous cells in our gastrointestinal tract as are present in our entire spinal cord. And as I've mentioned, it harbors a huge microbial ecosystem that is beginning to be recognized like another organ of the body. It's sort of like the liver in some ways, or the pancreas in some ways, in terms of its potential importance. Now, in the gastrointestinal tract, if you look at the abboral gradient, in other words, from the top of the gastrointestinal tract to further down, we find that there are a lot of microbes in the lower gastrointestinal tract. In the colon, we have about 10 to the 13th microbes per gram of intestinal luminal material. In the ileum, we have about 10 to the 7th. When you go up to the stomach, small intestine, there's a lot less. But with these regional differences, we are beginning to recognize that the different regions, the microbes in the different regions play different roles. So for example, in the ileum, they appear to play a major role in terms of the immunology, mucosal immunology of the gastrointestinal tract. And in the colon, they seem to play a larger role in terms of something called a bioreactor or metabolomic role. So what does this mean? Well, as far as the bioreactor or metabolomic role, some of this is illustrated on this slide. At the top, we have this insect called a termite. And if any of you live in a wooden home, this is something that we are very afraid of. Termites are insects that can digest a very complex carbohydrate called wood. Wood is a complex carbohydrate that has linkages that are very difficult to be broken by usual chemical techniques, at least in the human gastrointestinal tract. But in the termite, they have lignases that can break down some of these complex carbohydrates, but also have a microbiome that is able to break down these complex carbohydrates. Now, some of us may be very fat, some of us may be very thin, or somewhere in between. And it's been found that individuals who are very obese have a different microbiome than those who are very thin. And it's thought that some individuals who tend to be more on the obese side have a set of microbes in their gastrointestinal tract that tend to be more efficient in terms of breakdown of carbohydrates. There are many other factors involved as well, but that is one of the factors. If you take some of the microbes from an obese individual and put them into or inoculate germ-free mice early in their lives, the inoculum from the obese individual ends up helping the mouse develop into this obese individual, whereas the inoculum from the thin individual does not cause obesity. So the microbes in our gastrointestinal tract can certainly affect whether we are obese or whether we are thin. The immunologic role is also important. In the early 1960s, at the University of Chicago, there were some studies that were being done in conventional versus germ-free mice. So the conventional mice were just raised in regular cages, whereas the germ-free mice were raised in very highly germ-free conditions. And if you look at the axillary lymph nodes of the conventionally raised mice, you can see these germinal centers are very nice and big, whereas in the germ-free animals, they seem to be very wimpy looking, very small compared to these conventional centers in the axillary lymph nodes. So this is telling us that stimulation of the immune system by these microbes appears to also stimulate the development of the lymph nodes. In our gastrointestinal tract, we have about 7 different cell types, and here's the ileum on the left side, and here's the colon on the right side. And some of these cells include the enterocytes. These are the digestive absorptive cells. But we also have other cells that are involved in the innate immune barrier. Goblet cells, for example, produce mucus, and this immobilizes microbes so that they are not able to translocate into through the gastrointestinal tract. We have these cells at the base of the crypt called Paneth cells that produce defensive molecules. And these defensive molecules are very important in terms of protecting the highly mitotic region of the crypt here, because if you lose these centers where the cells are highly mitotic, you begin to lose the regeneration of cells in the intestinal epithelium. And they no longer are born in this region, and they no longer migrate to the tip of the villus where they are extruded. So this is very important to have these. And these are all responsive to certain types of microbes in the gastrointestinal tract. Now let's go back in history a little bit. And here we have this insect called Drosophila melanogaster, which I'm sure you're aware of is also sometimes called the fruit fly. And work that was done in Germany in Christian Nusslein Volhart's laboratory earned her and her colleagues the Nobel Prize for developmental biology of the Drosophila. What they did was they found that there were certain mutants of Drosophila that if you placed their proteins on a gel compared to wild-type Drosophila, you saw different patterns. And the day that one of her graduate students noticed this difference on the gel of a mutant fruit fly versus a non-mutant fruit fly, she came to talk with Professor Nusslein-Volhart, and Professor Nusslein-Volhart looked at the gel and said, "Das ist toll." Now, if any of you know German, toll means awesome, cool, really neat. So these proteins received the name toll proteins. Several years later, In the 1990s, Professor Charles Janeway at Yale University did some work looking at macrophages. And he found that in macrophages, that there were certain proteins that were linked to the microbes in a particular niche. And they responded to these particular microbes and then found that these proteins were very similar in nature as to those found by Professor Nusslein-Volhard in her studies in Drosophila. So these became the toll-like receptors, and now we know that there are approximately 12 of these toll-like receptors present in various cells of our body. And so, for example, we have toll-like receptor 4. Toll-like receptor 4 responds to lipopolysaccharide, which is present in very high quantities in gram-negative microorganisms. And this toll-like receptor 4 causes a response in the cytoplasm of these cells, and this response causes a dissociation of nuclear factor kappa B from an inhibitory factor that allows the nuclear factor kappa B to go into the nucleus and act as a transcription factor to produce certain kinds of cytokines, immunologically active molecules. And the same holds true for many of these other toll-like receptors. Now, studies that were done by the group that was at Yale University, Professor Medjetov, who had worked with Professor Charles Janeway, in 2004 published a paper in Cell that was very important, that showed that toll-like receptors, when they are present in a gastrointestinal epithelium, and if there are microbes present in the gastrointestinal tract, if you subject that intestine to a mild injurious agent, almost nothing happens, okay? There's homeostasis of the intestinal surface and robust repair in response to injury. If you have a mouse that's a knockout of a toll-like receptor 4 or toll-like receptor 2, and you subject that intestine to some injury by mild soapy-like material, there's enhanced sensitivity to injury and diminished capacity to induce repair. In other words, these mice, within 1 week, started to develop a hemorrhagic colitis and started to die. So that's the knockout of the toll-like receptor. But what happens if you have the toll-like receptor present and you don't have stimulation of the toll-like receptor? So they did this, they tested this by treating the animals with 4 different antibiotics, and that got rid of these toll-like receptor ligands, the microbes in the gastrointestinal tract. And then they subjected the gastrointestinal tract to this soapy-like material, and they found that the same thing happens as if you had a toll-like receptor knockout. Within 1 week, those animals became, had a hemorrhagic colitis and started to die. Now, this is telling us something very important. This is telling us that TLR ligands and signaling are crucial for the intestinal surface to protect and repair itself in the face of infectious or inflammatory insults. So in other words, tickling of these toll-like receptors by bacteria is important in terms of development and also normal repair of the gastrointestinal tract. Now let's go back also in history, and here are 3 individuals who won the Nobel Prize. The 2 guys on the left here, Bruce Butler and Jules Hoffman, won 25%, and Ralph Steinman won 50% of the Nobel Prize. Well, I'm going to focus on Ralph Steinman's work, and Ralph Steinman's work involved these interesting looking cells here. These are cells that are found in different parts of the body. They are called antigen-presenting cells, and they are of the monocyte macrophage lineage, and in certain parts of the body, they are also called dendritic cells. In the gastrointestinal tract, these dendritic cells have the capability to send up these arms or dendritic processes from the subepithelium into the lumen of the intestinal tract, and they're able to capture microbes, and that changes the surface of the dendritic cell. And the interaction of the dendritic cell with undifferentiated lymphocytes, depending on what they are exposed to in the lumen of the gastrointestinal tract, tells these undifferentiated cells to differentiate into either T effector cells or tolerizing T cells. This is very important because if you have an imbalance of effector T cells versus tolerizing T cells, you either have the inability to fight infections, or you may have not enough tolerance to self, where you begin to attack your own cells of the body. So, for example, you develop autoimmune diseases if you do not have the tolerizing capability. So it's thought that these are very important in terms of that particular balance. And as you can see here, there's this interplay with the types of microbes that are present in our gastrointestinal tract. Now, let's talk a little bit about the microbial ecosystem in the human. And where does this begin? And when does this begin? Is there a fetal microbiome? And this is a quote from an article that was published in 2012. And the first sentence in the discussion section says, "Every one of us enters the world devoid of microbial colonization because of the sterile environment of the womb." Is that true? Is the womb actually sterile? We are also taught as neonatologists that meconium is sterile. We are taught that the baby's skin right at the time of birth is sterile. So is that really true? Well, we are beginning to recognize more and more that perhaps this is not as dogmatic and true as as we thought it once was. This is work that was done at Stanford University by Drs. Dan DiGuglio and colleagues, and they were able to take some amniotic samples, amniotic fluid samples from Dr. Roberto Romero's laboratory. Dr. Roberto Romero is the head of the National Institutes of Perinatal Health at he has his laboratory in Detroit, Michigan. And he has many amniotic fluid samples that he has saved over time. And so looking at several different amniotic fluid samples at from different gestational ages, they saw that the bacterial DNA had this linear relationship, so that if you had a gestational age at delivery that was relatively low, there were more bacterial cells or microbial DNA in the amniotic fluid than if the gestational age at delivery was high. If the gestational age at delivery was high with this low level of microbial DNA, they also saw low levels of IL-6, interleukin-6, and white blood cells in the amniotic fluid. Whereas these over here, where the gestational age was low with high levels of microbial DNA, they found lots of IL-6 and also considerable white blood cells, suggesting the possibility that perhaps premature labor may somehow be associated with the inflammatory response that comes from the higher microbial load in the amniotic fluid at an earlier point in time. Studies done at Baylor College of Medicine in Houston by Dr. Kirsty Agard involved looking at the placentas of humans. And she looked at over 320, she looked at 320 subjects and found that there was microbial DNA in almost all of these subjects in the placenta. And that there was also an association with the types of microbes they found and the level of maturity and delivery. And also they found that there seemed to be a relationship with the types of microbes they found and the types of microbes in the maternal oral cavity, which was of interest. Work that we did at the University of Florida involved evaluating microbes from meconium, and we did primarily premature babies. And we saw that there was a considerable amount of microbes in the meconium of premature babies. But the diversity was actually not very high, but we were able to detect fairly high quantities of microbes in meconium. So where does this come from? Well, it has potentially different sources, but here is one potential source, that they could be migrating from the maternal urogenital tract and the vagina and crossing the choriodecidual plate and getting into the amniotic fluid. And what does the fetus do during the last trimester of pregnancy? Well, it swallows large quantities of amniotic fluid every day. And so we found in our studies that about 50% of the microbes in meconium are also found in the amniotic fluid from these other studies that had been done at that time, suggesting that this meconium microbiome might be coming from swallowed amniotic fluid. Still only just a hypothesis. So we're beginning to recognize that there is a very strong possibility that the mother-infant dyad, the mother and the fetus dyad, is actually a triad. It's a holobiont, which involves an ecosystem that also involves a microbial component. This microbial component not only contributes to the immune system, but as I mentioned before, it also plays an important role in the metabolites that are released. So you could be getting metabolites from the fetus, or you could be getting metabolites from the maternal gastrointestinal tract, from the skin, from the mouth, from the urogenital tract, that could be active in terms of development of the fetus, and that may also have epigenetic potential. So just to summarize, where could these microbes in the fetal maternal unit be coming from? It's thought that the vagina might be one source. The maternal intestine might be a source where there's translocation of bacteria into her bloodstream or carried by certain cells like the dendritic cells, or also from the maternal oral cavity, which would be a hematogenous origin. Now, this whole area of the in utero colonization has become highly controversial. There have been several studies that have looked at this very carefully, and they have found that perhaps some of the microbes that have been seen in the studies of the placenta and the amniotic fluid may actually be due to background colonization rather than the colonization within the organ itself. Nevertheless, this is something that I think several studies now have begun to support more and more, that there is a non-sterile womb situation. But the story is still somewhat controversial, and I want to make sure that you all understand that. Now, if we have these two different paradigms, a sterile womb paradigm and in utero colonization hypothesis, with the in utero colonization hypothesis, with either vaginal or cesarean delivery, the baby is already born with having microbes present on the skin, in the gastrointestinal tract, etc. But in the sterile womb paradigm, what happens is that the baby picks up bacteria going through the cervical canal, but in cesarean delivery does not pick up microbes going through the cervical canal. And this has actually been studied, and I'll get to some of this a little bit later. But first, I want to just mention that even if we invoke a sterile womb hypothesis, the maternal gastrointestinal tract still contains microbes. And this is from a study published in Science in 2016. And this was done in mice. If you take germ-free mice that are colonized by a special bacteria called E. coli HA107, and you inoculate them on day 10, what happens is that by day 15 to 20, those E. coli die out. They do not persist in the maternal intestine. So when they compared inoculated to non-inoculated offspring over time, and these are the inoculated offspring over time, looking at two different kinds of innate immune cells, you see that in the control babies' immune systems, these cells are very low compared to those whose mothers were inoculated. And this is telling us that the microbiome of the mother's gastrointestinal tract is important in development of the immune system. Now, what about the metabolomic function during pregnancy? What about the microbes that we find in the stools of the pregnant woman? Could they play a role in terms of the health and subsequent development of the newborn? Well, this is a study that was done by Dr. Omri Koren and colleagues, published in Cell in 2012. And the question he asked was, what about the different trimesters of pregnancy? Are there different kinds of microbes in the maternal gastrointestinal tract? And he used these new sequencing techniques to determine this. And this is something called a principal component plot. And here we see T1 is trimester 1 and trimester 3. And we see these different circles here, which represent a different microbiome from an individual, okay? And these individuals, microbiomes from trimester 3 are different than those in trimester 1. And these here are all controls, males and females. Females are non-pregnant, but during pregnancy, we see that the microbes are different. And in the first trimester of pregnancy, they are different than in the third trimester of pregnancy. This is really interesting because if you look at the first trimester of pregnancy and the third trimester of pregnancy, something happens. There's an increased weight gain in the mother that's due to not only the weight of the infant, but also that mother generally gains more body fat and also becomes insulin desensitized. If you take the microbes from this first trimester of pregnancy and put them into a into a germ-free mouse, that germ-free mouse ends up being normal in adulthood. But if you do that from the third trimester of pregnancy, the mouse ends up having insulin desensitization and ends up being fatter. So these microbes from the mother in the third trimester of pregnancy actually cause this almost type 2 diabetes-like picture in the mouse. Again, suggesting the metabolomic role of microbes. Now let's get back to our C-section issue that I started to get to before. Now, in different parts of the world, there are different C-section rates. And here we see the C-section rate, okay. India is sort of in the middle. China, higher C-section rate. South America, very high C-section rate. North America, about 30%. Canada, somewhat lower. Parts of Europe, quite low. Okay, Scandinavia, only about a 20% C-section rate. United States, about 30%. So we have these different parts of the world that have different C-section rates. And in certain parts of the world, the C-section rates are high, not so much due to the need for having these C-section rates, but there are other reasons. Some of them perhaps due to convenience and other factors that lead to these. Well, we are beginning to recognize from an epidemiologic standpoint that those babies born by C-section are different than those babies born by vaginal delivery in terms of their development of autoimmune diseases and other problems. So how does this happen? How does this autoimmunity happen with C-section versus vaginal delivery? Well, here is sort of a paradigm. Here's vaginal delivery, where you have exposure to the vaginal microbes, and you have normal microbial seeding of the gastrointestinal tract and normal development of immunity. But with C-section delivery, you have lack of exposure to the vaginal microbes. And along with this lack of exposure to the vaginal microbes, you have abnormal microbial seeding of the gastrointestinal tract. And it's thought that this leads to abnormal development of immunity. Okay, well, why is it, do we have studies that show that this is different, that the vaginal delivery actually leads to a different microbial ecosystem? Well, this is a study that was done in Venezuela by Dr. Dominguez Bello. And this is a principal component plot, the type that I showed you before. And these blue circles all represent different microbiomes. And you see that there's a dark blue and a light blue. And the dark blue represents the baby's skin, and the light blue represents cesarean section. So there's a very strong relationship to the microbes found in the baby's skin at 1 week after birth to cesarean section. What about vaginal delivery? These pinks and the reds are the microbes of the baby at 1 week after birth and vaginal delivery. And what we see here is that these cluster together. And with vaginal delivery, we see vaginal microbes present in the maternal, in the baby's gastrointestinal tract. Again, supporting the hypothesis that we see over here. Several subsequent studies have been done, and this is one of the most recent ones by Shao and colleagues that was published in Nature in 2019. And the question that was asked in this paper is it only the vaginal delivery that was associated with the development of a different microbial ecosystem? And looking at these babies at different time points after birth, it was recognized that delivery mode did play a large role. But maternal antibiotics also played a large role. Stay in the hospital played a large role. Breastfeeding played a large role. So there are different factors that were involved in the acquisition of the microbes. So in terms of results summary here, the mode of delivery affects the composition of the intestine through the neonatal period in infancy. And C-section delivered babies harbored more hospital-acquired bacteria, and C-section delivered babies received more antibiotics. C-section babies also took longer to start feedings. So what this is telling us is that it's not just the vaginal delivery versus C-section that's important here. It's also the more antibiotic use that you see in C-section delivered babies. There's also a greater stay in the hospital in C-section delivered babies, and babies who are C-section do not receive the same amount of breastfeeding as do the vaginally delivered babies. So this is actually something that plays a very large role in terms of the types of microbes that end up being, that the baby ends up being colonized by. Now, with this situation about the babies born by C-section versus vaginal delivery, it's been thought that it's possible to partially restore the microbiota of C-section born infants via something called vaginal microbial transfer. So in other words, what is done by vaginal microbial transfer is that you take babies who are born by C-section and those babies have their mother's vaginal tract. You take a gauze swab and put that into the mother's vaginal tract, and you keep that there for approximately 1 hour. And then you take that gauze pad and put it into the baby's mouth after the baby is born by C-section, and swab it, and also swab the baby's face and the chest. And this is sometimes called a microbial baptism. Well, this study was actually, this was actually studied in a paper that was published in Nature Medicine, and they had a very small number of babies that they studied. 18 dyads, 7 were born by vaginal delivery, 11 by cesarean section. And 4 of those who were born by cesarean section were swabbed, and 7 were not swabbed. And what did they find? Well, here we see those babies who were swabbed or inoculated in the green, C-section delivered babies here in the red, and the vaginally born babies here. And these numbers, these histograms represent Bacteroides relative abundance. And here you can see that the inoculation actually led to microbes in the gastrointestinal tract, which were similar to those of the vaginally delivered babies. Very important, very interesting finding. So this told us that you could actually inoculate the babies using this technique. However, even though this seemed to be very exciting, the American College of Obstetrics and Gynecology and others gave a warning, and this warning is seen here, especially in the red. We recommend against the practice of vaginal seeding until better data are available, establishing the safety and benefits of this practice. Now, just think about this a little bit in terms of what this might mean. If the mother has a congenital herpes infection that is undetected, and you try to do this seeding into the baby, you could actually kill the baby by inoculating that baby with herpes infection. So this is certainly a concern. What about this disease that we call necrotizing enterocolitis? Does the microbiome play a role in necrotizing enterocolitis? Here we see a typical picture of a baby with a distended abdomen, some periumbilical erythema. And here's pneumatosis intestinalis, portal venous gas. This baby was taken to surgery, had this area of necrotic bowel. What is the pathophysiology of this disease? Well, we think that the microbiome does play a significant role, but we also have to recognize the fact that necrotizing enterocolitis is a lot more tricky than we had originally thought. There are many imposters to necrotizing enterocolitis. That's really not the topic of this lecture, but a very important component to remember is that we still do not have a really good definition of this disease. And if we are going to evaluate this disease in terms of any kind of a pathophysiologic mechanism, we need to have a very good definition. So what about these neonatal practices that we may be doing to babies, or premature babies, that might be associated with the development of necrotizing enterocolitis? So this is a pogo cartoon. We have met the enemy, and he is us. Okay, so here we have 2 different babies, one born at 23-week gestation, the other here born at 29 weeks gestation. And the question we have is, when would this baby develop necrotizing enterocolitis at corrected gestational age compared to this baby? Studies that have been done, and this is from a study that I did with Dr. Mohan Pami from Baylor College of Medicine just a few years ago. And this looked at the corrected gestational age in weeks at necrotizing enterocolitis diagnosis. And here we see that there seems to be a peak right around 29 to about 31 weeks. And so this baby here is going to develop it later. in terms of corrected gestational age compared to this baby. So it'll take 6 or so weeks for this baby to more likely develop it compared to 2 weeks for this baby. We also were able to collect sequence data from 8 different neonatal intensive care units that had looked at the microbiome in the intestinal tract of premature babies prior to the development of the disease. And here we see 2 sets of histograms looking at babies who developed necrotizing enterocolitis, seen on the right, and control babies, seen on the left. And these are relative abundance of certain phyla of bacteria. And here what we see on the left in the control babies in the phyla of bacteria is that we have the blue, which stays about the same, and this dark yellow, which stays about the same. The blue is the proteobacteria. And in the necrotizing enterocolitis babies, the blue increases over time. So this particular phylum of bacteria, the proteobacteria, are higher in those babies who develop necrotizing enterocolitis over time. And the yellow decreases compared to stability over here in the controls. And these are the firmicutes. Those are more the gram-positive microbes that we see associated more with the firmicute phylum, okay? And we also see a difference in these yellow bacteria over here, and we see the presence of these this yellow. These are the Bacteroidetes, and here's the histogram of the Bacteroidetes. Neck babies, they're considerably lower than. What does this mean? Well, the proteobacteria tend to be those microbes that have high lipopolysaccharide in their cell wall and stimulate the toll-like receptor 4 that stimulates the inflammatory response, as I mentioned before. The firmicutes, which we see actually decreasing in the necrotizing enterocolitis babies, are those more considered to be more like the good bacteria that produce short-chain fatty acids such as acetate, propionate, and butyrate. Butyrate, for example, is very important in as a fuel for the colonocyte. And butyrate is also important for keeping intestinal junctional epithelium together, preventing increased permeability or leakiness of the gastrointestinal tract. The Bacteroidetes are important because they produce a certain protein called PS protein, and this particular protein is involved in stimulating the anti-inflammatory response. And here we see that this is lower in the necrotizing enterocolitis babies, suggesting a pathophysiologic association between the types of microbes in the gastrointestinal tract and the development of the disease. Now, what about antibiotics? Here's a slide that shows the different antibiotics that are most common, or the different pharmaceutical agents or drugs most commonly used in premature babies. And way at the top are ampicillin and gentamicin. But we also see other antibiotics commonly used, cefotaxime, vancomycin, etc. Okay, so of the 10 top drugs used in the neonatal intensive care unit, 4 of them are antibiotics. How does this relate to the potential development of disease? Well, here on this slide, we see something called dysbiosis. We see here on the left side, panel A, a balanced microbial community. Here we see increased species diversity. And in general, when you think about ecosystems, high diversity is a good thing. This is an ecosystem where all the different components are working together to have a healthy microbial or a healthy systemic community. If you take antibiotics, then you lose a lot of the microbes, and you begin to have decreased species diversity. And with this, you have altered toll-like receptor signaling and immune dysregulation. And this is potentially problematic. This dysbiosis is potentially problematic. What happens when we use antibiotics, either in a mother or in a newborn? This is a study that was done by Arbiola and colleagues, and we see different kinds of microbes in mothers or infants that were not exposed to microbes. And here, again, we're talking about these phyla of bacteria that I mentioned before. When they are exposed to antibiotic, when the infants are exposed to antibiotics and the mothers are not exposed to antibiotics, we see more proteobacteria, less firmicutes. When the mother is exposed to antibiotics, we see a lot of proteobacteria. And the mother and the infant exposed to antibiotics, again, a lot of proteobacteria, very few of the firmicutes. So antibiotics affect the types of microbes present. And as I mentioned before, this could play a role in the development of intestinal inflammatory processes. Now, we also know that the intestine is the site that causes systemic inflammation. And if you have inflammation in the intestine, this inflammatory response does not just stay in the intestine, it goes to other organs as well. And so the lung, the brain, the kidney, various other organs can be affected as well in terms of this systemic inflammatory response. And we're beginning to recognize more that the types of microbes that we find in the gastrointestinal tract may play a role in the development of other disease entities, such as bronchopulmonary dysplasia, and even neurodevelopment by causing an increased inflammatory response. Again, this is an area that requires a lot more study, but we're beginning to recognize, and the hypotheses are being generated in this regard. Now, a lot of what I talked about just recently is associations, and associations do not prove causality. What do we need to prove causality? Well, I'm going to talk a little bit about randomized trials and multi-omics. I'm not going to talk that much about animal models. Now, what about antibiotic usage and the development of necrotizing enterocolitis? There have been several studies, and I mentioned in one of my previous slides, that there's an association with the development of necrotizing enterocolitis and the antecedent use of antibiotics. In fact, there's a 50-fold increase in risk of necrotizing enterocolitis if antibiotics are previously used. But again, this does not prove causality. So we need to have prospective randomized controlled studies. And a few years ago, we started to do this, and this is not very easy to do because of all the dogmas in neonatology of giving antibiotics early on. And this is a study that we recently completed, and we now have 2 publications and another one that is just about accepted that relate to this particular study. And we had 3 different groups. One group that needed to get antibiotics. So if the mother had a high fever or whatever, we gave those babies antibiotics. Another group was considered to be a low-risk group. The mother did not have any fevers. The baby was born by cesarean section, had no symptoms, looked really good. That baby did not get antibiotics. But then about 50 to 60% of the babies were in this group here that ended up being randomized to get antibiotics or not get antibiotics. So this was one of the first randomized controlled studies of antibiotic use in premature babies. And we had several different results here. And here I just want to show you the microbiome and metabolomic clustering. And here we see that this is group A, those babies who definitely received antibiotics, and these are the randomized control babies. This is the group that received no antibiotics. This is the group that received antibiotics. And you can see there's different clustering of microbes depending on antibiotic use in the gastrointestinal tract. The metabolites are also different, and this happens to be a metabolite that we got from the gastrointestinal tract called gamma aminobutyric acid. And the gamma aminobutyric acid in those babies who received the antibiotics was actually lower than in those babies who did not receive the antibiotics. And this also related to the types of microbes that we saw in the gastrointestinal tract. So this is very preliminary data on only 98 babies, and we are hoping that we have a multi-center trial started to look at this more closely in the near future. Now, what about multi-omics? And here I give a parable that maybe some of you are familiar with. This is the 6 men of Indostan. And I'm going to read this poem to you, this parable to you. The 6 men of Indostan, they were blind, to learning much inclined, who went to see the elephant, though all of them were blind, that each by observation might satisfy his mind. The first felt the side of the elephant. It feels like a wall. The second felt a trunk. It feels like a spear. The third felt a trunk. It felt like a snake. So you can see that these different men of Indostan are feeling different parts of the elephant. And so these men of Indostan disputed loud and long, each in his own opinion, exceeding stiff and strong, though each was partly in the right, and all were in the wrong. Are you getting the gist here? If you only look at one part, for example, the microbiome, or the metabolome, or what we are seeing clinically, this only gives a part of the picture. It shows an association. We need to have something that is much more mechanistic. So each type of omics data may provide a list of differences associated with the disease. Individually, they can be useful, but analysis of only one data type is limited to correlations, mostly reflecting reactive processes. We don't know whether the chicken or the egg came first, in other words, rather than causative ones. If you just look at these individuals, they can be hypothesis generating, but they don't really test causation. And to look at multiomics will give you a much better feel for the entire picture. And if you get the entire picture, this can more readily lead to disease treatment targets or prevention of disease. So here is just another way to look at this with different omics technologies. We have different features. For example, antibiotics, no antibiotics. We can look at enrichment analysis and look at these different, what is called network analysis. And we can see the different nodes, the different metabolites or different microbes that are expressed differently using this network analysis. And with this, we can get a lot more information than with single omic analysis alone. This can also relate to what we feed to our babies. And now I just want to go back to the early 1990s where we had this, what's called the nutrition pyramid. And this was developed by the United States Department of Agriculture. And you can see that there are different nutritional components that it was recommended to take. These are nutritional guidelines. But we are beginning to recognize more and more that each one of us is different than the other one of us. And we may need slightly different nutrients for us as individuals. And so we are now beginning to recognize that these pyramids or these guidelines don't really work so well. And the pyramid of the future can be based on omics. And this is a strategic plan that the National Institutes of Health has put out for the 20 to 2030 strategic plan. And what this involves is evaluating the different omics to have precision nutrition for individuals. And this involves different of the omics, for example, the genome, the transcriptome, the proteome, metabolomic profile, and the pathway identification that I spoke about before. And also microbial host interactions. Now, when it comes to the neonate, we also, especially the premature neonate, we are beginning to recognize that the 23-week premature neonate is not the same as a 32-week premature neonate. The needs are quite different. And personalized nutrition and being able to target the types of nutrition is going to be very important. And so using some new technologies, for example, severity scoring systems that we can determine by machine learning, and then applying the multi-omics, such as metabolomics, microbiome, inflammatory mediator analysis, and then looking at network analysis, we hopefully will in the future be able to determine nutritional, microbial, therapeutic, as well as preventative interventions and optimize nutrition for these babies rather than use a one-size-fits-all guideline-based approach. The tools we have for integration are seen here. Machine learning, mediation analysis, system network, etc., as well as the multi-omics. Teams are going to be important in terms of this approach. So I'd like to stop here with some take-home messages. Microbes play an important role in our overall physiology before and after birth. Perturbations in our microbial host interactions have lifelong and perhaps even beyond consequences. Well-done clinical studies along with newly developed multi-omic technologies will help us reach our goal of more personalized medicine. And I'd also like to acknowledge several people and universities and the National Institutes of Health and funding agencies that I've had the opportunity to work with in the past. And I'd also like to thank you for your attention.
Thank you, Professor Dr. Joseph Liu. And we had such a myriad of impressions from your lecture, how microbes and multi-omics technology is very useful not only during pregnancy but during birth process and also later on in our practices of neonatal care. And we have plenty of questions for you, sir. At least as I see now, 16 questions as of now. Shall we take the first question for you?
Certainly.
And Dr. Chandrakala will also be coordinating the other questions. The first question to you, sir, from Dr. Bharani Anand. He wants to know that could necrotizing enterocolitis be a clue to primary immune deficiency? Can I repeat the question?
Yes, I understand. Can necrotizing enterocolitis and our understanding of the pathophysiology be a clue to immune deficiency? I think that there are some possibilities. Whenever you're learning about the immune system, whenever you're learning about the inflammatory system, it provides another piece of the puzzle. And so I think, yes, it can. But as a major player in terms of immune deficiency, I think that there may be, it might be a little bit of a leap. But yes, whenever you find another piece of the puzzle to immunity, I think that it can be very helpful. Yes.
Okay, the second question is, so we do see intra-abdominal calcification both in the antenatal period and postnatally as an incidental finding. And do these babies need follow-up? Now that you are saying that meconium is does have microbe. So do we just leave them, or do we need to do follow?
Well, I think that the kinds of microbes that we find in meconium tend not to be the pathogenic microbes. They tend to be the commensal microbes. And we have to remember that the vast majority of microbes that we live with are the commensal or friendly microbes. And so our gastrointestinal tract, our skin, they're filled with these different microbes. And so I guess that I would not necessarily be concerned if the baby is otherwise healthy to do any kind of specific follow-up. Just good common health follow-up in those kids, but not anything special.
Yeah.
Okay. Chandrakala, would you take the next question?
Yeah, sure. There's another question from Dr. Akum Toshi. So, is there any role of starting probiotics in cesarean section babies? Is it beneficial? So,
There have been some studies. There have been some studies that have suggested that there may be some beneficial role, but then there are other studies that have been done that have sort of countered those. I think that the jury is still out. Now, the problem with probiotics is there are about 200 different probiotics, okay? And when you talk about probiotics, you have to qualify which probiotic you are talking about, okay? You have to look at the studies that have specifically looked at that particular probiotic agent. And I think that that's one of the major pitfalls of what we do with probiotics, is that we are putting them all into the same category, that they all do the same thing. It's sort of like antibiotics. Chloramphenicol is very different than ampicillin, is very different than vancomycin. So we have to consider the different probiotics as having different roles. And I think that this is a field that we have to be very careful in terms of moving forward, because especially when we're talking about premature babies, when we are talking about babies whose gastrointestinal tracts are actually not highly colonized, but you are putting something into that baby's gastrointestinal tract that is potentially even keeping other microbes, other commensal microbes, from developing diversity. I just think we need to be very careful with that whole that entire area.
Okay, sir. Thank you. There's one more question from Dr. Manoj. This is regard to those babies born of cesarean section. What percentage of babies become bronchiolitis, or they will have bronchoplasma later in life?
Well, it is thought that there is a higher prevalence of asthma. I don't know about bronchiolitis, but there is a higher prevalence of asthma in those individuals who are delivered by cesarean section. Some of the epidemiologic studies show that. Again, whether the cesarean section itself is causative, or whether the antibiotics or the lack of breastfeeding or the longer stay in the hospital with cesarean section may be involved with this, I don't think we can pin any one of those specifically to greater asthma or autoimmune diseases, etc.
Okay. So there's a question by Dr. Rani. What is your practice regarding withholding of feeds during Paxil transfusions?
I don't hold feeds during Paxil transfusions. I think that the more recent studies have very strongly supported that there is no need to hold feedings, and the evidence is just not there for holding feedings during Paxil transfusions.
So, Madam, would you ask other questions?
Yeah. Actually, there are some more questions on necrotizing enterocolitis, sir. Use of probiotics for reduction of incidence of necrotizing enterocolitis.
Yeah, so this is a lecture in itself, or maybe more than one lecture in itself, but just to try to make things very brief. There have been several meta-analyses of studies that suggest that probiotics may be helpful in reducing necrotizing enterocolitis. These meta-analyses were all done with different probiotic preparations, okay? And this is a major issue. There is only one prospective randomized trial of a probiotic, of a single probiotic that was done in UK that was adequately powered for necrotizing enterocolitis. And it showed no difference in outcome in terms of sepsis or necrotizing enterocolitis. Okay. They also did not see any detrimental effects in that study, but again, no beneficial effects. There are other studies being done right now on probiotic preparations that are prospective, randomized, but these are studies that have to be done very carefully. And I think that perhaps the future will give us more information on that. So at this juncture, I think that the evidence is still somewhat weak for an individual probiotic preparation in the prevention of necrotizing enterocolitis.
So the next question regarding necrotizing enterocolitis, how do we do the practice of antibiotic use for management of necrotizing enterocolitis with the current knowledge of omics?
So as far as if you are strongly suspecting necrotizing enterocolitis in a baby, I think that at this juncture, I would not change practice markedly because we still have a lot to learn. But I think that we are learning very rapidly now because we are beginning to recognize that a lot of those babies that we have diagnosed as having necrotizing enterocolitis may actually have something else. And some of the newly developing technologies, such as ultrasound, bedside ultrasound, can help us. This is not yet fully developed, but I think that this is sort of the wave of the future where the bedside ultrasound techniques might give us a lot more information of what the babies actually have, whether they have true pneumatosis intestinalis, portal venous gas. And my hunch is that in the future, what we're going to be doing is a lot of these cases that we have called medical necrotizing enterocolitis will no longer be getting 7 to 10 days of antibiotics or 7 to 10 days of NPO as we have done in the past. But I don't have a strong recommendation as far as changing practice right now. But I think that this is going to be something that is evolving. In the next few years, we will see some real changes.
The next question is on oral iron administration and associated intestinal dysbiosis. So what's your current thinking on that? Administration of oral iron and intestinal dysbiosis.
I think that that's a very, very good question. Studies that have been done by Dr. Nancy Krebs at University of Colorado has actually shown that high iron intake may actually lead to some dysbiosis. And so the question is it possible that some of the iron that we are giving to our premature babies or all the blood transfusions that cause iron overload may actually play a role in dysbiosis? This has not yet been adequately studied, but I do know of some people who are actually studying this and have been able to garner grant funding for this. So this is an excellent question. I wish I had a really good answer for you. Very likely it does play a role, but we are going to find out more in the future with these ongoing studies.
Yeah, this was asked by Bharat Srivatsa. And the next question is, what is the role of IL-6 and IL-8 in the diagnosis of early onset sepsis? IL-6 and IL-8?
Well, they are both inflammatory mediators. Certainly, IL-8 is a very important chemokine. You find it in fairly high quantities with intestinal inflammation. One of the problems is that some of these cytokines are very short-lived. And so they may not be as sensitive or specific as we would like them to be. So I don't think that we have a really good handle on how we can use IL-6 and IL-8 in the diagnosis of intestinal inflammation. But they certainly do, they are increased with intestinal inflammatory responses.
Chandrakala, you want to take up the other questions?
So there's an interesting question here by Dr. Sridhar. How much similarity will there be between breast milk microbiome and vaginal microbiome? Will it be a good alternative to vaginal seeding to use colostrum or breast milk to wipe the baby?
Well, I think that it would probably be better to just feed the baby breast milk. So if there's breast milk available, just have the baby eat from the mother, rather than I think that or the this technique where you take small, even just a couple drops of the breast milk and put it into the baby's mouth early on. There have been some studies shown that there's a benefit to doing that in terms of development of the immune system. The mechanisms are still not very well understood, but there appear to be some benefits and no downsides to that anybody has seen.
Fine. There's one more question. Can we think of inoculating gut microbiome of healthy infant to at risk of neonates in near future? This is by Dr. Vijay.
So, in other words, like doing a fecal transplant, is that, or a fecal implant?
Yeah.
It's possible. It is possible. And there is one study that is ongoing right now. And it has not yet been published as a full study to my knowledge, but they're looking at the potential for fecal transplants in babies born by cesarean section. So taking fecal samples from the mother and doing the same thing as the vaginal seeding that I mentioned before. The results of that study, to my knowledge, are not yet in.
Okay. There's a question. How important is the role of microbiome in NEC in term babies associated with asphyxia and congenital heart diseases? Is this condition really NEC?
My guess, I don't think we have the final answer on that. And there are studies being done with babies with congenital heart disease. I think that there's probably a role, but with congenital heart disease, some of the problems that we see in the gastrointestinal tract are actually somewhat different than what we see in a routine preterm baby. For example, with left-sided obstructive lesions such as interrupted aortic arch or hypoplastic left ventricle, there's a lack of blood flow to the gastrointestinal tract. And that lack of blood flow is very different than the pathophysiology that we see in our premature babies that is largely dependent on microbiota-related inflammatory response. So yes, I think that there probably is a role, but whether it's a major role, I think it's less than in the preterm baby.
Okay. Okay. There's a question by Dr. Hariram from Bangalore. He's a leading neonatologist here. And
he's a good friend. He's also a good friend of mine.
Yes, he appreciated the talk, sir. And is there any correlation between delayed passage of meconium in extreme preemies and NEC? This is his question.
As far as that being documented as a really strong relationship, I would say no. That But when you think about the possibility of a baby who has poor motility, a newborn with very poor motility, there could be an association. But whether that's causative, I don't think that it's causative, but it could be an association. Those babies who do not pass meconium that readily, those babies also could have poor motility, which can cause microbial overgrowth and inflammatory responses in the intestine and potentially necrotizing enterocolitis.
Okay. Madam, would you like to take further?
Yeah, there is one question from Dr. Saurabh Datta. The whole metagenomic analysis gives the abundance of phyla as relative abundance, but the absolute abundance may be more important. Are there ways of assessing absolute amounts?
Yes, yes.
Yeah.
Simply by doing targeted PCR. You can target the PCR for a particular microorganism and then take the entire microbial DNA load as your denominator, and then you have your target PCR for a particular microorganism. Yes, that can be done.
Yeah. And do you recommend the moms to receive probiotics to improve the gut microbiome in the baby?
Well, similar to my other answers before, I think the jury is still out. I do not recommend the mom to take probiotics. No, I think a good diet, if she wants to eat certain foods, yogurt, for example, that contains microbes, I think that that is probably a very good thing to have a mixed microbiota and to eat foods that may have some fermentation products is not a bad idea, but I would not, I don't think that we have a really strong need for recommending that at this time.
Okay. Another related question with the mother: maternal zinc levels and relationship with immune status of the offspring. Maternal zinc levels.
Sure. Well, zinc is known to be an important cofactor in many immune processes, and to have a normal zinc level and to not be zinc deficient is a good thing. Okay, so I think that to maintain a normal zinc level, to eat foods that do contain zinc is very reasonable. Sometimes a multivitamin preparation that also contains minerals is very reasonable.
The next question is whether Omics panel could be used for screening in a sepsis screen in a newborn. How far can it be used?
Ask me 5 years from now. This is we still don't have that. We certainly still don't have that, but I think that this is one pathway we can go toward screening, not just for the presence or absence of a microbe, but also the body's response. As you know, you can harbor all kinds of microbes, but it's not until the body responds to it that actually causes damage. And so multi-omics may play a role there, but we don't have it yet.
Okay. Dr. Chandrakumar from Chennai has asked this question. Is umbilical cord a good source of microbiome, and could it be potentially useful in transferring favorable microbes to other infants with unhealthy microbiome?
Not to my knowledge. As far as the umbilical cord microbes, I don't think that there has been much done in terms of microbiome in the umbilical cord. So I don't have an answer to that. I don't think so.
Okay. And another question, a very curious question. Should we use probiotics if the baby is being bottle fed?
Gosh, everybody comes back to the probiotics question.
I would rather discourage bottle feeding rather than giving probiotics, but your answer, please.
I think that one can actually develop a microbiota by skin to skin care, by having close contact between the mother and the baby. And so I don't think that it's necessary to put a probiotic into formula to promote health. I know that several companies have done this already. The evidence that tells us that this is actually beneficial is very sparse. So I'm very skeptical if that's going to actually play a role in health and disease in the future of the babies that are getting the probiotic and formula. But absolutely, I agree with you. I think that there is a microbiome in human milk, and that's where I'd like to have the babies getting their microbiome is from their own mothers.
Okay.
Yes.
So there are some more questions. Would you like to continue? Let's
do a couple more. We can do a couple more. Yeah.
Okay. Then Chandrakala take the rest.
You can go, ma'am. No problem. Maybe 20 minutes we finished. Maybe one or two questions we can take, ma'am. You can go on.
Nalinikant has asked some question. You can take that question. Shall I finish it? Or you will do
finish it up, ma'am. Finish
it. Would you use CD64 for diagnosis of NNEC?
To my knowledge, this has not been used as a diagnostic measure for NEC. So I have not used it. No, but I'm not saying that it's not useful. I just don't know.
Okay. So what is your experience of CMPI in newborn with bovine milk derived fortified with exclusive breast milk?
CMPI, would you explain that to me, please? Cow's milk, protein intolerance?
Cow's milk protein intolerance.
In terms of what? Repeat the question, please.
It's a bovine milk derived fortifier with breast milk fortification. So fortification of bovine milk derived fortifiers. What is your experience of CMPI?
So I think that some of the cases that we are calling NEC are likely due to bovine milk, bovine milk proteins. And those, you can actually see cow's milk protein intolerance or food protein induced enterocolitis from human mothers who are taking high dairy, And so if we are suspecting that in a breastfed mother, we actually suggest that the mother decrease her or stop her dairy intake, which is sometimes very, very difficult. We also sometimes will take a extensively hydrolyzed or amino acid formula in some of those babies and try that and see if that will ameliorate the symptoms.
I think we may give these two last questions for you, sir. Does freezing or storing breast milk, especially in human milk banking, does it alter the microbiomes?
So in regular freezing conditions in -20, there's very minimal alteration of the microbiome, at least according to the studies that we have done. So it's not that bad to freeze the milk. But you do lose some of the other components, such as cells, and you may kill off some of the enzymes. So it's not just the microbes that we are worried about. It's some of the other components as well.
And one more question. Glycerin enema given as a routine in the Japanese neonatal units and intestinal microbiota. Your comments, please. Glycerin enema.
I don't do it. I very, very rarely give glycerin suppositories or glycerin enemas. I'm not saying that it's never done, but it's very rare. I do not do it routinely. I know that in some neonatal intensive care units, this is routine. I don't think that this has ever been shown to be of benefit in terms of prevention of necrotizing enterocolitis, in terms of prevention of feeding intolerance and other issues. So no, I don't do that routinely.
One last question, sir. Can we give microbiome administration to the mother? Can we prevent prematurity?
So this is a really interesting question. It has not been adequately studied, but I think that it's a hypothesis that if you provide certain kinds of microbes to a mother, and in the oral cavity or in the gastrointestinal tract or in her vaginal tract, can that prevent prematurity? I think it's a question we have not answered, but I think it's a tenable hypothesis.
Thank you, sir. Such an excellent interaction. I think it lasted for more than 35 minutes, which is almost equivalent to your talk time. That is the reaction you have created among the 200 participants from various countries. Really, we enjoyed the evening. Hopefully, you also did enjoy. And over to Dr. Manoj and Chandrakala.
Thank you, sir. Thank you.
Well, all good things need to come to an end. This is what we keep saying during our webinars every time, the same sentence, because we can't stop. 20 minutes is good enough, but then we can't tax you more. Thank you so much, Professor New. May I now request my colleague, Dr. Ravindra Varma, to propose the official vote of thanks and close the session. Over to Varma. Please unmute yourself.
Thank you, Dr. Manoj. On behalf of IAP Neocon, it is my great pleasure to propose the vote of thanks this evening. First of all, our thanks are to Dr. Joseph New, who, in spite of a busy schedule, could spare his valuable time for this webinar. We are extremely grateful for his wonderful and illuminating talk. Thank you, sir, for joining us today. Dr. Kumbuda and Dr. Chandrakala were kind enough to accept our invitation to moderate this session, and we are eternally grateful to them for their valuable time. Thank you, madam.
Thank you. Thank you for your invitation.
Finally, just like any conference, no webinar is a success without the people attending it. We are thankful to all our attendees from various countries. There were more than 250 attendees at some time when I was just seeing. You are our strength. Thank you very much. Finally, we welcome you all, including, of course, Dr. Joseph New, for our next webinar on PDA in preterms on 28 November, Saturday, 7:30 p.m. Thank you all. Good night, good day, good afternoon, good evening. Thank you very much.
Thank you.
Bye-bye. Thank you.
Bye-bye.
Bye-bye.
Thank you.
Bye.